Okay, so our next speaker is Teresa Lee. Her talk's entitled Longevity and its Transgenerational Inheritance is Enabled by H3K9 Methylation.
Hello. First, I'd like to thank the organizers of this meeting for giving me the opportunity to talk to you about my work. In the CATS lab, I've been very interested in how chromatin is either maintained or reprogrammed between generations, and I study this in the context of longevity, which is a very complicated process in which there are many inputs, both internal and environmental, that interact to affect one physiological outcome, which is the lifespan of an individual. So worms were the first organism in which it was determined there was a true genetic component to aging. And more recent work has also demonstrated that there is a further regulation beyond just DNA sequence at the level of chromatin. So this indicates that aging is also epigenetically regulated. Now, we all know that in the nucleus, DNA is maintained as chromatin, in which is wrapped around a protein core to form a nucleosome. And this nucleosome has tails to which modifications can either be added or removed. So to focus you on the two modifications that I'm going to concentrate on for my talk, I'm showing methylation of histone 3 at lysine 4 and lysine 9. So H3K4 methylation is associated with open chromatin and active gene expression. In part, this is added by the compass complex, of which WDR5 is one member. It's removed also by the histone demethylase SPR5 in worms. Conversely, H3K9 methylation is associated with closed chromatin and gene repression, and again, in part, it's added by MET2 in C. elegans, which is specific to the germline. And so I just want to draw your attention to a key that I have in the corner, in which I'll use green to represent active chromatin and purple to represent repressive chromatin for the rest of my talk. And so a few years ago, the Brené lab at Stanford discovered that removing the compass complex from mutant animals extends lifespan. So again, compass adds the active chromatin mark H3K4 methylation, and WDR5 mutants are defective in the compass complex and have a longer lifespan. So that's represented in this diagram by the size of the worm. Not only are WDR5 animals longer lived, but they're able to pass this longevity onto their descendants, which the Brené lab demonstrated in a follow-up paper. So to highlight how crazy of a phenotype this is, really, these animals in the generations I've highlighted here are genetically identical to wild type, and yet they are as long-lived as WDR5 mutants are themselves. So they somehow maintain the memory that an ancestor of theirs was a WDR5 mutant and presumably had lower amounts of H3K4 methylation. And so this posed an interesting conundrum to us, because if longevity and its inheritance is caused by a reduction in this active chromatin mark here, why wouldn't the phenotype revert as soon as there's a wild-type copy of the gene present in the genome? So the fact that we see longevity persisting for several generations indicates that there's something actively blocking the inheritance of, rather the reversion of longevity immediately. And we have some recent work from our lab that hints at what this might be. So we have demonstrated that between generations, chromatin must be reprogrammed, and we've shown that this occurs in two steps. So first, the demethylase removes H3K4, the active mark, and this reduction in gene expression is reinforced by the activity of MET2, which adds repressive H3K9 methylation. To examine what happens when we play around with the inputs in this system, we decided to examine a WDR5 mutant, which has less of the active H3K4 methylation. And we were curious about what would happen to the readout here when we examined H3K9 methylation in these mutants. We demonstrated that there's a dramatic increase in H3K9 methylation. So using chromatin immunoprecipitation followed by qPCR, we examined several genomic loci. Here I'm showing you 4 germline expressed genes. And you can see that in wild type in black and the negative control of MET2, there's low enrichment of H3K9 at these loci, but in dark gray, WDR5 mutants have a drastic enrichment of this repressive chromatin mark. So WDR5 animals have less active chromatin and a great increase in repressive chromatin, and this posed a model that we wanted to test, whether this repressive H3K9 mark could both confer longevity and allow for its inheritance. So to do this, we first set out to recapitulate the original observation. Here I'm showing you a survival curve, which is how we measure the lifespan of a population of individuals. So on the x-axis here, you see the fraction of the population that remains alive with the days in the assay shown, on the y-axis is the fraction alive, and the days is on the x. So we begin our assay with living animals, so all of them are alive, but as individuals begin to die, you see the curve descending gradually until here the last individual dies at day 29. So since WDR5 was shown to be long-lived, we'd expect the curve to be to the right of the wild-type curve. So WDR5 animals die later than their wild-type counterparts, and this trend remains true for the entirety of the assay. However, when we examined WDR5 mutants, we found that they were actually shorter-lived than their wild-type counterparts. So WDR5 mutants in our hands died earlier than wild-type, and this trend also remained true for the entirety of the assay. So you see that their survival curve is actually to the left of wild-type. And this was confounding to us and more than a little worrying because it conflicted with what had been previously shown. But we followed our WDR5 mutants for many generations, assaying lifespan frequently. And we found that lifespan in this population gradually increased until it did become long-lived. So here what I'm showing you is our transgenerational experiment. What I'm calling P0 is the first homozygous mutant population after WDR5 had been maintained as a het for 5 generations. So, as I've alluded to, short-lived early generation WDR5 animals, we see that the loss of the activity of WDR5 hurts lifespan in some way. But over several generations, we see that this curve creeps rightward until it does not look any different than a wild-type animal. And then after 18 or so generations, we see that lifespan continues to increase, and these animals become long-lived. So the curve is entirely to the right of wild-type. I'm using a cartoon to illustrate what we saw again. WDR5 begins short-lived, looks normal-lived, and then attains longevity after 18 or so generations. And I just want to highlight how drastic the change we observed in this population was. So again, early generation WDR5 mutants are very short-lived, and over many, really dozens of generations, we see that longevity increases in this organism until we were eventually able to recapitulate the original increase, which was about 30% longer than wild type. And so that begs the question of what could possibly be accumulating in these animals over this amount of generational time, right? Because this is 20 or more generations. And again, these are all genetic clones of each other. We also know that this isn't due to background effects in the genome because I've done this 2 other times and seen similar results. Our previous work raised the possibility that it could be repressive H3K9 methylation that's accumulating in this population. So we used ChIP-qPCR to look at an early generation short-lived WDR5 population, shown in blue, and a late generation long-lived WDR5 population. And our preliminary data, here I'm showing you 2 loci at which we knew there should be enrichment for this mark. And we see that late generation mutants have a much higher enrichment than the early generation WDR5 mutants. And so this leads to a model in which repressive chromatin accumulates over time. So in a wild-type animal, we believe that there's a certain balance in the genome of active chromatin and a lower amount of repressive chromatin. WDR5 mutants don't have this active chromatin mark, but they're hurt because they have low amounts of H3K9, and they're therefore not normal. Over time, eventually, H3K9, in the absence of this active mark, appears to accumulate in the genome until it reaches a certain point that it becomes protective for lifespan, and that's when we see the appearance of longevity in this population. But if this is true, we have a genetic test that we can do, which is we would presume that the enzyme that adds this repressive H3K9 mark would be required for longevity in the WDR5 mutant population. So when we examined MET2 mutants, we see that they are in fact much shorter lived than their wild type counterparts. And here again, I'm showing a mid-generation, slightly long-lived WDR5 mutant. So this indicates that the activity of MET2 is required for longevity, or at least for a normal lifespan. When we then examine the double mutant, we see that MET2 is able to suppress the longevity we'd expect to see in a WDR5 animal. So this again strongly indicates that H3K9 is required for the longevity we see in the WDR5 population. So this model of longevity in WDR5 mutants also points to a way in which the longevity itself can be inherited, because we've demonstrated that H3K9 may be accumulated over many generations. So I've told you already that the Brunei lab demonstrated that WDR5 mutants can pass their longevity phenotype down to their descendants, in which the wild-type descendants of WDR5 ancestors, shown in orange, are as long-lived as their ancestors themselves. And so if H3K9 is part of the mechanism that allows for the inheritance of this phenotype, we would expect that removing MET2 in these descendants would again suppress the inheritance. And in fact, that's what we see. So here in pink, I'm showing you descendants of WDR5 mutants from which we've removed MET2. And you can see that they are shorter lived than wild type, and certainly shorter lived than their cousins, which are simply wild type descendants from the same WDR5 ancestors. And so to put our entire model together, we believe that, again, in a wild type animal, there's a certain balance between what is active and what is repressive in the genome. And in a way, this acts to limit lifespan. In the absence of the activity of the compass complex, we see that animals are hurt initially, but over time they accumulate enough repressive chromatin in their genomes to which we see the appearance of longevity in this population. Once they've attained longevity, they are then able to pass this on to future descendants, even though these descendants have wild-type copies of the genes present. However, since they have a wild-type copy of WDR5, they can then begin adding active chromatin back in their genome, and eventually we see that the balance of chromatin is reset to normal levels, and we see a reversion of the longevity phenotype after several generations. So overall, we've demonstrated that the ability to properly reprogram your chromatin between generations is crucial for a normal progeny. And the failure to do so can have very drastic consequences, affecting even a complicated trait like longevity. We've also demonstrated that the inheritance of this repressive chromatin mark, H3K9 dimethyl, is what enables both the appearance of longevity in WDR5 mutants, as well as the inheritance of that very complex trait. And so to end, I'd like to thank my advisor, Dave Katz, as well as Amanda Engstrom, a graduate student who's been helping me with this project. I'd also like to thank the lab for making it a really fantastic place to work. And I'm grateful to Bill Kelly and the CGC for strains. And I'd like to acknowledge my funding sources, the NSF, as well as FIRST, which provides my postdoctoral fellowship. And thank you for your attention.
I'm wondering how you can exclude the possibility that the inherited agent is actually a small RNA, and those small RNAs are the ones that then have an impact on the chromatin state?
Yeah, that's, we have not excluded that possibility, and we have also not even examined that possibility. So this very well could be that small RNAs are involved, but we do know that at least one readout will be the inheritance of H3K9 dimethyl.
Right, but that would just be a secondary consequence then.
Yeah,
so
that's very possible.
The agent that determines it, it's a readout of another transacting factor, right?
Yes, that's very possible, and I think it would be interesting to investigate whether whatever the factor is needs to be inherited maternally or paternally, because you can imagine that that can affect the amount of generations it could be inherited for. It seems less likely to me that small RNAs could be inherited for as many generations as we've shown in our transgenerational experiment, but it's certainly possible, especially if there's some sort of amplification step involved.
But it's not really a transgenerational experiment. I mean, you're just seeing that phenotypes become worse over time. And you could easily envision that this is because you're starting to accumulate over time more and more small RNAs that are responsible for your effect.
Yes, and yet the only, so many things get passed along to the next generation in the oocyte. And so one of those things could be a small RNA, which would be a more direct effect. So we have ways to investigate that.
Thank you.
I wonder if you found any other mutant, any other compass mutant, or other long-lived mutants that act similarly to WDR5 in a transgenerational phenomenon.
Yeah, that is something that we are considering. So the Brunei lab examined all three components of the compass complex in worms, and we have not recapitulated this effect in those other members of the compass complex. And there's just many other chromatin factors that could be involved, and so we're beginning those investigations now, but we have not confirmed any of them. Thank you.